Hi guys, welcome to Chapter 17, Part 2. We're going to talk about translation, mutations, and how, while not every organism goes through transcription and translation the same, genes are universal. Alright, so let's start with talking about translation. Last time we talked about transcription, which was DNA to RNA. So now we're going to RNA to synthesizing a polypeptide or protein. Alright? So, the translation of mRNA to protein can be examined in more detail, which is what we are going to do. Alright, molecular components of translation. A cell translates an mRNA message into a protein with help of transfer RNA or tRNA. Molecules of each tRNA are not identical. Each carries a specific amino acid on one end. Each has an anticodon on the other end. The anticodon base pairs with a complementary codon on the mRNA. Okay. Alright, this figure on page 337, this shows the basic concept of translation. As a molecule of mRNA is moved through the ribosome, here's your molecule moving through your ribosome. Alright, codons are translated into amino acids one by one. The interpreters are tRNA molecules, each type with a specific anticodon at one end and a corresponding amino acid at the other end. The tRNA adds its amino acid cargo to a growing polypeptide chain when the anticodon hydrogen bonds to a complementary codon on the mRNA. The figures that follow will show more detail on how this works. So this is your mRNA strand. Each set of 3 are your codons. Your anticodons match to those, and that is what signals your amino acid to be released to go on to your polypeptide chain. Okay? Alright, a tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long. Flattened into one plane to reveal its base pairing, a tRNA molecule looks like a clover leaf. Okay? Here's showing that clover leaf flattened into one dimension. Alright, this is on 338 if you want to read more about what's going on here. This two-dimensional structure shown here, your 4 base paired regions and 3 loops are characteristic of all tRNAs, as is the base sequence of the amino acid attachment site at the 3' end. The anticodon triplet is unique to each tRNA type, as are some sequences in the other 2 loops. Okay? The asterisks mark bases that have been chemically modified, which is a characteristic of tRNA. Okay? Alright, the bottom figure here, this 3-dimensional structure, is showing what that clover leaf, when it's non-flattened, what it looks like. And it's showing the symbol that's used in the book instead of showing you that 3-dimensional double helix. It shows it as almost like a little tube with your anticodons at the end. Alright, it's cleaner to look at in your figures. Anticodons are conventionally written 3' to 5' to align properly with your codons that are written 5' to 3'. Alright, for base pairing, RNA strands must be antiparallel, like DNA. For example, anticodon 3' to 5' AAG pairs with the mRNA codon UUC. Okay? Remember, there are no T's now because we're dealing with RNA, so thymine has been replaced by uracil. Okay? Alright, this is up close of that 2-dimensional structure, up close to your 3-dimensional and our symbol for the book. Alright, because of hydrogen bonds, tRNA actually twists and folds into that 3-dimensional molecule. It's roughly an L shape once it's in its 3D state. It looks like a clover flattened, but when those hydrogen bonds are in effect holding that 3D shape, it does kind of take on an L shape. Okay? Accurate translation requires 2 steps. The first step, a correct match between tRNA and an amino acid, is done by the enzyme aminoacyl tRNA synthase. Alright? And then second, the correct match between the tRNA anticodon and the mRNA codon. Flexible pairing at the 3rd base pair of a codon is called a wobble, and it allows some tRNAs to bind to more than one codon. So with that flexible base pair and that third base pair, it's flexible. The wobble, it allows it gives it more options. Okay. All right. This figure, also 338. This is your amino acyl tRNA synthase enzyme. All right. This is showing the active site that binds the amino acid and the ATP. There's your ATP. Okay, there's your active site. Alright, in this figure, the ATP loses two, it gains one, it loses two phosphate groups, and joins the amino acid as AMP. Okay? Alright. Appropriate tRNA covalently bonds to amino acid, displacing AMP. Okay. And then your tRNA charged with its amino acid is released by the enzyme. So this is how the linkage of that amino acid to your tRNA, it's an endergonic process that occurs at the expense of ATP. ATP loses those two phosphate groups and becomes AMP, adenine monophosphate. Okay? So this is how that first step, your tRNA gains its amino acid. Alright? Ribosomes, they facilitate the coupling of your tRNA anticodons with your mRNA codons in protein synthesis. The two ribosomal subunits, your large and small, are made of proteins and ribosomal RNA or rRNA. Okay? This figure on 339, the first is showing your computer model of a functioning ribosome. This model is of a bacterial ribosome showing its overall shape. The eukaryotic ribosome is roughly similar. Ribosomal subunit is an aggregate of ribosomal RNA molecules and proteins. Notice you can see the large and the small subunit. Okay. These bottom two are schematic models showing the binding sites, and once those binding sites are in action. The binding sites, a ribosome has an mRNA binding site and 3 tRNA binding sites. Here's your mRNA, there are your 3 tRNAs. Okay? This schematic represents how the ribosome will appear in later diagrams. So this way you're familiar with what it's showing you here. Okay? This last little figure here is showing it in action with the mRNA and tRNA working through the ribosome. Your tRNA fits into a binding site with its anticodon base pairs with an mRNA codon. The P site holds the tRNA attached to the growing polypeptide site. Alright. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide chain, and discharged tRNA leaves from the E site. So A site is where your next to be added comes in. E site is your growing amino acid, or no, P site is the growing amino acid, and E site is exiting. It's leaving out, okay? Here's up close on those models again. Alright, a ribosome has those 3 binding sites we just talked about for tRNA, the P, A, and E sites. P site holds the tRNA that carries the growing polypeptide chain. If you look back at this figure, P site is in the middle. So P site is your growing chain. A site holds the next amino acid to be added. So A site is coming in. E site is the exit site where your discharged tRNAs leave. So once it gives its amino acid to that growing chain, it leaves out of the E site. Okay? There are 3 stages of translation. There are 3 steps to building a polypeptide: initiation, elongation, and termination. All 3 stages require protein factors that aid in the translation process. Alright, the initiation stage of translation brings together mRNA, a tRNA with the first amino acid and the 2 ribosomal subunits. First, a small ribosomal subunit binds with mRNA and a special initiator tRNA. Then the small subunit moves along the mRNA until it reaches the start codon, which is AUG. proteins called initiation factors bring in the large subunit that completes the translation initiation complex. Okay, this is showing this mRNA binding site. So mRNA binds to the binding site of this small ribosomal subunit. Your initiator tRNA reads this mRNA strand until it locates AUG, which is always amino acid MET or methionine. Alright? And that binds, and that brings in the large ribosomal subunit, and that initiator becomes the growing polypeptide chain in your P site. Okay? So the elongation part. During elongation stage, amino acids are added one by one to the preceding amino acid. Each addition involves proteins called elongation factors and occurs in 3 steps: codon recognition, peptide bind formation, and translocation. Okay? This figure, which is on page 341, it's figure 17.18. Alright, this first step is showing codon recognition. The anticodon of the incoming amino acyl tRNA base pairs with the complementary mRNA codon in the A site. Okay, so it comes in and pairs in the A site with its complementary codon. Hydrolysis of GTP increases accuracy and efficiency of this step. Alright, step 2, there's your GTP. Step 2, the peptide bond formation. The rRNA molecule of the large ribosomal subunit catalyzes the formation of a peptide bond between the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. So this step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site. Okay, so your P site has your current polypeptide chain. This rRNA molecule catalyzes the release from this tRNA and joins to the amino end of this new amino acid. So your chain, your polypeptide chain, is moving from this tRNA to this tRNA. This tRNA will now move to your P site. Okay? So now see the polypeptide chain has been transferred to this newest amino acid. Your first tRNA is now empty. Okay? And it will then go through translocation, which is where it's essentially ejected, but where the ribosome translocates the tRNA in the A site to the P site. So A site moves to P site, and the tRNA, the empty tRNA in the P site moves to the E site, where it's released. mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site. So as this occurs, it's sliding the mRNA through to be read. Okay? Take your time, go back and reread over that figure, and this should make sense. If not, let me know. Alright? So now we've initiated and elongated. Now let's terminate this translation. Termination occurs when a stop codon in the mRNA reaches the A site of the ribosome. The A site accepts the protein called releasease factor. The releasease factor causes the addition of a water molecule instead of an amino acid. This reaction releases the polypeptide, and the translation assembly then comes apart. Okay, so here is your release factor. It read that stop codon. Oops, there it is. Alright. This is on page 342 if you'd like to see it. So when a ribosome reaches the stop codon on the mRNA, A site ribosome accepts a release factor, a protein shaped like a tRNA instead of an aminoacyl tRNA. Okay? Now, secondly, what occurs here is the release factor promotes hydrolysis of the bond between tRNA in the P site and the last amino acid of the polypeptide. thus freeing the polypeptide from the ribosome. So your polypeptide is now free from your ribosome. And in your third step, your two ribosomal subunits and the other components of the assembly disassociate. So everything comes apart. So once your polypeptide chain has been released, the rest of the assembly comes apart, ready to go on to the next thing. Okay? So polyribosomes. A number of ribosomes can translate a single mRNA simultaneously, forming a polyribosome or a polysome. Polyribosomes enable cells to make many copies of a polypeptide very quickly. This figure here, also on 342, this is a micrograph that's showing a large polyribosome in a bacterial cell. Okay, there are your ribosomes all synthesizing mRNA. Okay, all right, completing and targeting the functional protein. Often translation is not sufficient to make a functional protein. Polypeptide chains are modified after translation. Completed proteins are targeted to specific sites in the cell. Alright, during and after synthesis, a polypeptide chain spontaneously coils and folds into a three-dimensional shape. Proteins may also require post-translational modifications before doing their job. Some polypeptides are activated by the enzymes that cleave them. Other polypeptides come together to form the subunits of a protein. Alright, targeting these polypeptides to specific locations. Two populations of ribosomes are evident in the cells: your free ribosomes, which are in your cytosol, and your bound ribosomes, which are attached to your ER. Your free ribosomes mostly synthesize proteins that function in the cytosol, and your bound ribosomes make proteins of the endomembrane system and the proteins that are secreted from the cell. Ribosomes are identical and can switch from free to bound. Okay, polypeptide synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the ER. Polypeptides destined for the ER or for secretion are marked by a signal peptide. A signal recognition particle, or SRP, binds to the signal peptide. The SRP brings the signal peptide and its ribosome to the ER. Okay, this figure 343 is showing that polypeptide synthesis begins on a free ribosome in the cytosol, as your free ribosome, as your SRP binds to the signal peptide, halting synthesis momentarily. The SRP binds to the receptor protein in the ER membrane. The receptor is part of a protein complex that has a membrane pore and a signal cleaving enzyme. Okay, here the SRP leaves and the polypeptide synthesis resumes with its simultaneous translocation across the membrane. The signal peptide stays attached to the translocation complex. Alright, in this figure, the signal cleaving enzyme cuts off the signal peptide, and then the rest of the completed polypeptide leaves the ribosome and folds into its final conformation. Okay? That's when they're targeted to the ER. Okay? Alright, now we've talked about translation. We've gone through transcription and translation. Now let's talk a little bit about what happens when this doesn't go exactly according to plan. Alright, you have mutations. So, let's start with point mutations. They can affect protein structure and function. Mutations are changes in the genetic material of a cell or virus. Point mutations are chemical changes in just one base pair of a gene. So point mutation is where one base pair has been changed, and that affects the entire rest of what that sequence is coding for. So the change of a single nucleotide DNA template strand can lead to production of an abnormal protein. Okay, this showing wild-type hemoglobin DNA and mutant DNA. This one base pair here has been changed. It's one point in the change. When your mRNA is transcribed and translated, normal hemoglobin gives you GLU. Okay? Mutant, when this one base pair has been changed, makes your mRNA transcribes differently to your mRNA, which translates differently to your amino acids and gives you valine, which is the sickle cell hemoglobin. That one base pair is the difference in normal versus sickle cell hemoglobin. Okay, you have types of point mutations. Within a gene, they can be divided in two general categories. There are base pair substitutions and base pair insertions or deletions. Alright, so this figure, if you'd like to see it larger, is on 345. Let's examine it up closer here. Alright, this first strand is showing you your template strand for your wild type and what is usually produced. This here is showing you a silent point mutation. A, it's a substitution. A is put in instead of G. That one pair has changed. And so you end up with a U instead of a C. However, it did not affect the amino acid sequence. So that's a silent mutation. Okay? Now this one, which is a missense mutation. Alright? A missense mutation is substitutions that change one amino acid to another one. Alright? It may have little to no effect, or it could change your amino acid completely. And that, just depending on what it's building, could change what your entire protein is trying to do. Alright, see, this one just completely put a T instead of a C. And so that changed the rest of the coding and changed your amino acid. This is a nonsense mutation. Alright? And nonsense mutations is a point mutation that can change the codon for an amino acid into a stop codon. So what that nonsense does is wherever that nonsense mutation occurs, it changes the sequence of amino acids into a stop codon. And so that stops your DNA from being transcribed or translated any further. Okay? It will not be translated into amino acid because it reads it as a stop and it thinks it's the end. Alright? Your frame shift mutation. Frame shift is where, let's see, make sure I explain this correctly. Alright, your frame shift is insertion or deletion of nucleotides that alter the reading frame. And so your triplet grouping has been changed. So this occurs whenever the number of nucleotides inserted or deleted is not a multiple of 3. So it pushes everything, either pushes or slides back together the entire reading frame. So if this had one base pair insertion, adding an A where there wasn't one before, when that's translated, this one happened to have translated into a nonsense codon and stopped it. Okay? It's a nonsense mutation, it stopped it. It may have smaller effect or it may have a lot larger effect. This one went straight to the nonsense. Now this one caused extensive missense. This one base pair being omitted changed every single amino acid past that point because it shifted every base pair one to the left and it changed that set of 3. So now everyone was being read at a different place, so it completely shifted the rest of that protein. Okay? This one has a complete amino acid missing. This had 3 base pairs deleted. Again, changes what your protein turns out to be. It shifts that entire amino acid out of the system. For this one, it's lysine. Took it completely out of the polypeptide chain, so now that protein is lacking that amino acid. Okay? So, what's going on with all these? A base pair substitution replaces one nucleotide and its partner with another pair of nucleotides. Silent mutations have no effect. produced by the codon because of the redundancy in the genetic code. Missense still code for an amino acid, but not necessarily the right one. Nonsense change an amino acid codon into a stop codon, nearly always leading to a non-functional protein because it cuts it off too soon. Okay? Insertions and deletions, or additions or losses of nucleotide pairs. They have a disastrous effect on the resulting protein more often than substitutions do. So substituting may or may not completely change the polypeptide you're aiming to receive. However, insertions or deletions, they are going to change from that point forward, so they are much more harmful to that protein sequence than just a substitution. That substitution may affect one amino acid pair, where an insertion or deletion are going to affect every amino acid from that point forward. Alright, insertion or deletion may alter the reading frame, which produces a frame shift mutation. That's where from that point forward it's altered. Okay? Spontaneous mutations can occur during DNA replication, recombination, or repair. And you have mutagens, which are physical or chemical agents that can cause mutations. Okay? And our last section, 17.6. Alright, while gene expression differs among the domains of life, the concept of the gene is universal. So your archaea, which are prokaryotes, but they share many features of gene expression with eukaryotes. Bacteria and eukarya differ in their RNA polymerases, termination of transcription, and ribosomes. Archaea tend to resemble eukarya in these aspects. Bacteria can simultaneously transcribe and translate the same gene. In eukarya, transcription and translation are separated by the nuclear envelope, like we talked about earlier. In archaea, transcription and translation are likely coupled. Alright, this is just showing on page 347. This is showing that coupled transcription and translation in your bacteria. So as soon as your RNA polymerase, your polyribosome, is created as your DNA is transcribed, okay? It goes right on the order of your DNA strand. It's translated to mRNA and then transcribed into your polypeptide chains right sequentially, okay? So what is a gene? Let's go back to this question for just a second. The idea of the gene itself is a unifying concept of life. We've considered a gene as a discrete unit of inheritance, a region of specific nucleotide sequence in a chromosome, and the DNA sequence that codes for a specific polypeptide chain. Alright, this figure is revisiting everything we've talked about in this chapter. It's on page 348. I highly recommend you go back and take a look at it and make sure you understand the parts of what it's discussing. Alright, in summary, a gene can be defined as a region of DNA that can be expressed to produce a functional product, either a polypeptide or an RNA molecule. Okay, so this wraps up transcription translation. Excuse all these as I go down. Alright, make sure that you look over these. You should now be able to questions. We will discuss these more in depth. Make sure you answer your checkpoint questions. If there's anything that you don't understand, make sure that you ask. Make me a list if you need to. Let me know if you need any help, guys. I look forward to discussing this with you. Thanks. Bye.